Hello everyone, my name is Ettore, and today with Craig I'm going to talk to you about steerable neural cellular automata. So this is a follow-up from our original paper, Growing Neural Cellular Automata, where some colleagues and I show how we can, from a single seed, generate patterns that are very complex and able to regenerate from damage and be stable. But notice this interesting property, which is that every time I regenerate the pattern, it always comes out with the same rotation and reflection. And this might not be something that we want. And the cause of this is because these neural cell automata are anisotropic. And by this I mean that their behavior is dependent on their orientation, on the x and y coordinates, which is something which we might not want. But where is it coming from? Well, it's very easy to inspect, and it's due to the way they perceive the environment. They perceive the environment by using Sobel filters, Sobel kernels, which are the way that you can perceive gradients of the states of your neighborhood. In this way, you have two filters, one for the x coordinate and one for the y coordinate, that essentially breaks the isotropy immediately and makes them anisotropic. This does not mean that they cannot rotate. In fact, we already show how we can rotate them in the original paper. But this is done by giving an extrinsic angle theta, which then we use for in a rotation matrix to rotate the perception field. And in this way, they can rotate patterns. But notice that this is not a property of the cell, it's actually a property of the environment. It's like this, the grid has changed and the cells react accordingly. And we might not want to do this. Maybe we really care about not having an extrinsic coordination, or we do care about rotation and non-reflection invariant. What can we do? Because after all, like, biological cells are largely isotropic and use other means to break symmetries, such as noise, material gradients, light, gravity, and who knows how many other methods. So 2 years ago, Alex, Eivind, and I published a paper on ALIFE called Differentiable Programming of Reaction Diffusion Patterns, where we were already using isotropic neural cellular automata in a certain sense, because reaction diffusion is isotropic by construction, and we were using it on textures, and the results were interesting, but of course they were still, let's say, not as nuanced as we managed to do later on. But it was still an isotropic approach, and this was done, for example, essentially because the reaction diffusion does not use Sobel filters. Another paper which we published last year, also on ALIFE, was growing isotropic neural cellular automata. Here, we showed how we can generate patterns that are growing from a single seed or multiple seeds, able to generate things of all possible rotation and reflections. And the way it was done is by using not Sobel filters, but using Laplacian filters, which are isotropic by construction. But what if we wanted to have a specific fixed reflection? So, like, we do care about having a specific reflection, or the problem is not the anisotropy or isotropy per se, but it's that we actually don't want to have this extrinsic information. We want cells to organize themselves, and they can have their own orientation, but it just has to be themselves deciding it. Well, this is what we're going to talk about with growing steerable neural cellular automata, where in this case you will see that we will generate lizards of all possible rotations, but always the same reflection. So how do we perceive, though? Because since, like we were saying, that the kernels, Sobel kernels were the problem before, and they resolved it with Laplacians, what do we do now? Well, we will still actually use Sobel filters, but the important part is that we will use them in a way that is not dependent on any extrinsic data. And we'll show two variants of this principle. The first is probably the simplest to understand, is by using angle-based rotations, where simply, as opposed to having an angle which is given from outside, every cell has an internal orientation. They cannot perceive this orientation, but their perception is based on this angle. What they can do is they can rotate themselves. So they can decide to rotate left or right. And this makes them chiral or steerable. So they are not isotropic, but they are rotation invariant. Another variant is the gradient-based variant, where this looks a bit more complex, but the core principle is that orientation is an emergent property of the collective cells. So a cell by itself has zero orientation, but if it's nearby other cells, then they might have an orientation. And the way they decide that is by having a specific state, which they use to generate a gradient field. And then, based on how the state varies between your neighbors, you decide to orient yourself in a certain direction. So this means that if all your neighbors would have the same state, you would actually not perceive any gradient of states for all the states, not just the concentration state. And if you're wondering why we would care about this, it's because it solves some problems that we explore more into the paper, but we don't have time to talk about here. But even if we have now, let's say, some steerable neural cellular automata, which are only rotation invariant and not reflection invariant, how do we train them? After all, we need to break some symmetries, because like in isotropic NCA, before in isotropic NCA we had rotation invariant and reflection invariant, but now we are rotation invariant, and this means that all possible rotations would be fine for a loss perspective. So how do you decide that? Like in isotropic NCA, neural cellular automata can break symmetries by themselves because they are asynchronous. And this means that every single cell has a different kind of computation with respect to their neighbors, just by random chance, which makes them capable of accumulating noise that then can break symmetries. But then we still need to train them. So we will show two ways. One is by using rotation invariant losses, and the other one by using structure seeds, which is the one that Craig will discuss. So let's start with single seeds and rotation invariant losses. So here you can see how we start from a single seed, and the cells are very, very quickly breaking symmetries and deciding on how to coordinate themselves. But again, they will have all the same reflection, even if rotations are random. So how do we train this? Well, if in the original Cellular Automata paper, we were comparing a ground pattern towards a certain target pattern and say how much are you different from this one. We do this, but for all possible rotations of the target pattern. So all 360 degrees rotations, and select the minimum loss among them, and then backpropagate through that. And there's a fast way to do that with fast Fourier transform, but that's an implementation detail. The important part is that we say you get a pattern, compare it to all possible rotations of the target pattern, and backpropagate the loss with respect to the minimum loss that you find with this. If you're familiar with isotropic NCA, you will notice that this is strictly a simpler approach than what we were doing for isotropic NCA, because there we had to care about all rotations and reflections. Meanwhile, here we only care about all rotations. But notice that just like in isotropic NCA, training this is hard. And the problem is that if you were to compare the target pattern with all rotations of itself, you would notice that, of course, at 0 degrees angle, the loss is 0 because it's comparing itself with the exact copy of itself. But the more you rotate it, the more it changes the loss. And you notice that at 180 degrees, there's a very strong local minimum. And this would make it so training would almost always converge to a situation where you have a pattern which is a mixture of head and tail at both ends. And this is not nice, but we can solve this by adding an extra channel, which we augment the pattern with, which just says, on the north, I want all of this extra channel to be all zeros, and on the south, I want it to be all ones. And this changes the loss landscape, making this 180 degrees much, much smoother, and therefore able to be trained. Finally, we can also look at the angles of the results. You can see that angle-based variant and gradient-based variants have very different behaviors. The angle-based variant have a spiral at the center, and then they are all pretty much aligned afterwards. But spirals are not possible in gradient-based variants because their orientation depends on the orientation, well, the concentration of their neighbors. So the concentrations can only move can only vary smoothly. And therefore, what can happen is that you have, for example, first an orientation towards the right, then the angle gets completely suppressed and you don't perceive gradients of any direction, and then you start perceiving gradients on the left. And now I'll let Craig continue.
Hello, my name is Craig. Today I'll talk a little bit about how, as an alternative to using a specialized loss function, we can also structure our seeds to remove ambiguity for the model during training and direct its behavior out of training, similar to what we did with isotropic NCA. In fact, here, since we're only resolving ambiguity along a single axis, we'll see that we can use 2 seeds for steerable NCA as opposed to 3 needed to direct the isotropic model. Even more, we can achieve this direction with a fixed target and the same pixel-wise L2 loss used to train the original anisotropic NCA model with no additional auxiliary channels. Just like with the single seed experiments, though, you'll notice here that the reflections are always the same, regardless of orientation, as a result of the fixed Sobel perception filters. We'll explore these examples a little more later, but here this demonstrates how we can direct rotation of growth using just structured seed placement. In order to define initial conditions which effectively restrict the behavior of steerable NCA, we just need to take a bit of care in how and where we initialize the seeds. In the case of the lizard here, each seed is placed to serve as a point of origin for one of 2 subregions of the resultant pattern. By fixing the arrangement of the seeds during training and distinguishing them by color, we provide steerable NCA with this clear axial cue that it can exploit to construct the target pattern. Given that this is the only directional cue it has, the model performs exactly as we'd expect, and it learns to use these seeds as a framework for self-orientation. The seeds here roughly correspond to the upper and lower halves of the lizard, and as these 2 regions grow, at some point they intersect. It's at this point that the cue becomes apparent to the model, and it's able to orient each region to match the target. Since the model depends on this point of intersection to self-orient, we can rotate the seed out of training and observe the resultant pattern rotate respectively, just like we did with isotropic NCA. But what if instead of rotating the seed structure, we adjust the diameter? Doing this gives us a bit of a clearer picture in how the model depends on these border cell states at the point of intersection. Changing the diameter, after all, changes what these states are. And while the model is pretty robust and able to recover a stable pattern, we can see, especially here in the top 2 examples, how the model fails to settle on a direction when the seeds are too close. On the other hand, placed too far apart, like in this bottom left example, the 2 regions begin pattern formation before they've even had a chance to intersect and align with one another, resulting in this sort of 3-headed lizard where both regions attempt to complete the entire pattern on their own. Because isotropic NCA with its Laplacian perception filter is completely rotation and reflection invariant, its training necessitated a structured seed with at least 3 non-collinear points. This way, we define 2 axes about which the model can rotate and reflect. With steerable NCA, however, like we mentioned, the reflection is automatically given to us by the Sobel filters. So our seed structure can be simplified to just 2 points defining a single axis along which the model aligns its rotation. It is important to note that while we can use any number of seeds to train steerable NCA, our model is strictly chiral due to the invariable perception filters, so we can't simply use 3 points to attempt to manipulate the reflection of steerable NCA like we did with isotropic NCA. This highlights a major difference between the 2 architectures, where in steerable NCA, the cells can adjust their angle of perception, but just like isotropic NCA, they are still bound by its construction. Whichever reflection of the target is used during training is the reflection we're stuck with, and while this might pose an issue in the isotropic sense, this is pretty characteristic of the chiral systems we're attempting to model. Also, like before, we can visualize the perception of each cell as a vector field and see snapshots before, during, and after the model determines its orientation. Here, the cells are initialized at random angles, and as the 2 regions intersect, the angles stabilize to form this flow-like field depending on how the seeds are arranged. By adjusting the rotation of the seed structure, all we're really doing is adjusting this point of intersection, and consequently the relative angles of the constituent vectors in the flow field. With that, returning to our mosaic here, having trained the model on a fixed non-rotating target pattern, We can easily manipulate steerable NCA's rotation out of training just by appropriately adjusting the structured seed used during training. So thank you all for listening, and please let us know if you have any questions or comments. Thank you.